First, I'd just like to introduce everyone here. I'll talk a little bit about the project, but first I want to talk about the people. When I was hired to work at the Soil Health Institute, this group was already hired, and I hadn't met them. I didn't know anything about them. Boy, was I nervous coming into an institute where there was this huge project going on, and I had no idea what the scientists were like, but I had no reason to be nervous. This is an amazing team, and I haven't even done much with this project because they just do it all. It's pretty cool. So I'd like to just introduce everyone. Everyone met Shannon last night. This is Shannon Cappellazzi. I said it right. Good job. And each of these scientists are located in a different spot of the United States because they're focusing on sampling in those areas. Next is Kelsey Hoegenauer and Charlotte Norris and Paul Tracy. Paul's the manager of the project. Mac Bean. Where's Mac? Oh, yeah, sorry. I did it opposite. And Dan. And then Liz and Michael Cope. Yeah, Liz Rieke and Michael Cope. And the other neat thing about this team, they each chose the part of this talk that they wanted to give, and they all have different expertise, so we have a really broad range of expertise in this group. And it's been amazing. And now I'll show you the numbers, and you guys can be as impressed as I have been. So this project is our strategic approach for evaluating soil health indicators. Everybody is asking, well, what do you measure when you measure soil health, right? We all measure different things, all the soil scientists in the room. But what we wanted to do is just look at, take a strategic, comprehensive approach to look at how these soil health indicators actually behave across North America. And this project, specifically that we'll be talking about now, is part of a larger project. And Pipa talked about her part in the project, the TNC's part, and Shafali talked about the Soil Health Partnership. And those are our partners in this is TNC and Soil Health Partnership. And the project is funded by the Foundation for Food and Ag Research, and also by General Mills. So that's our collaborators. That's the big idea. So there's the NOLA look, and then there's Soil Health Partnership working on the farms, and then we're kind of doing the big long-term site assessment. So our approach is to measure and evaluate, look at these long-term research sites with long-term management, and look at how these targeted soil health practices or soil health measures are associated with both the management and the agronomy and the risk associated with these areas. And to start off this project, the first thing that had to happen was that we had to choose 31 soil health measurements. And I think, so there was a blue ribbon panel that came in, experts in all sorts of measurements of soil properties, physics, chemistry, microbiology. And the Soil Health Institute has published these 31 soil health measurements at different tiers. I did not intend for anyone to have to read this slide. And also, you don't even have to be able to get to that website. Just go Google and type in Soil Health Institute tier soil health measurements, and it'll be the first hit. Okay, but I just wanted you to know they're there and they're published. The next thing that happened is the Soil Health Institute brought in some panelists. I'd like to say they're experts. Most of them were, but me, because I didn't do a lot of plot type experiment, but it was a great opportunity to learn a lot more about plot experimental design and things like that. And we worked for 2 days. So first thing that happened is there was a call that was put out for volunteers to volunteer their long-term research sites. And they filled in information. There was a website form to fill out. And then this team was brought in to look at about 154 site applications. And we worked for 2 days going through all the basics of these site applications, trying to figure out, which sites we sample, which ones are appropriate, and which ones fit the experimental design. And the way we prioritize these sites is we first looked to see if the experiment had soil disturbance. Well, first of all, they all had to be replicated, and most of them needed to be replicated. But the priority was soil disturbance, soil surface cover, plant diversity. Those both kind of fall under cover cropping and systems rotations. We looked at crop nutrient amendments, so organic versus manure applications versus organic agriculture, and then traditional applications of fertilizer. We looked at crop and soil management. Geographic location, we wanted them quite distributed around North America. And then we also looked at, are these sites plugged into larger networks? Are they long-term sites of measurements of anything else? Also in this, so that was the priority of the kind of treatments we want. And then in the criteria to evaluate them, again, we wanted a pretty wide geographical distribution. We needed there to be replication in treatments, information on, we looked at the experimental design. There was a minimum of 10 years of management change in the area. So that's what, when we say long-term experimental sites, we're talking about 10 years or more of this management treatment. And then there were some sites that didn't have replication because they were really, really old historical agronomic sites. And we just included them because how can you just turn down sites with, decades of management history? And this is what that looks like. This is the spread across Canada, Mexico, and the United States. So 154 sites were submitted for consideration. Our official line is that we selected 120 for sampling, though that number kind of goes up and down a little bit. And what's the progress today? Well, we've determined the indicators, as I've just talked about. We've selected the sites. Of course, we've hired all the scientists that did the sampling. We've contracted with laboratories to do the analysis of the 31 indicators. We held a PI workshop, I think it was in January in Chicago. I just know it was cold. But look at this group of people that came. That's the photo of everyone that attended. And this was during the government shutdown, so that did not even include USDA sites. So what happened is we ended up having a webinar for the USDA cooperators. Now, this is the coolest part. Of 120 sites that were selected, they were all sampled this spring. So in that spring window, even though there was all this rain and many, many farmers didn't get their crops planted, we got the sites sampled. And so we're a total of a little over 2,000 experimental units that were expected or planned to be sampled, and we got 93% of them sampled. The rest will be sampled in the fall, and of course we weren't able to get to them due to logistics of weather. And the laboratory data we'll expect to have by the end of the year. So with that, we're going to let Shannon go, and we won't be introducing anyone since they've been introduced, and they'll just start talking about their part of the project. Thanks, Shannon.
Thank you. All right, so I'm going to give an overview of the protocols that we used, particularly while we were out in the field and doing our sampling. And as Cristine mentioned, we had a meeting with a lot of our site cooperators in, I think it was February, and that's where we kind of got through a lot of the logistics and talked about when we were going to get out to their fields and sampling. And there was just a huge amount of effort that was put forth from our cooperators, and none of these protocols, none of this would have been possible without their help and the cooperation. Most of the time we got out to those plots, it was early in the morning, and we had to do kind of a plot assessment. We met with these cooperators, made sure that we knew where the plots were, and then we went out to each of those individual plots and put flags in the ground where we wanted to have samples. Because at each of these locations, we had people who came out to the field to help us. There were people who wanted to come and help sample soils all day long. And so we needed to make sure that we had consistent protocols across this entire North American effort. So we put flags in the ground, and we trained the people who were helping us. Sometimes it was 1 up to 5, 6 people who were out there putting this whole effort together so we could get these 2,000 samples done in the spring. A lot of times for me, I would start by setting up the saturos. I know that Mac's going to talk more about these, but this is a method of measuring the saturated hydraulic conductivity, which for a lot of people can be a really time-consuming task. This machine put forth by the meter group can give you very nice, consistent results in a relatively quick period of time. It takes about an hour to 2 hours to run each of them, depending on a lot of things. So what I like to do when I get out to those sites, I would put the saturos in the ground, and I normally had 3 of them with me. So I would set up 3 different saturos like you see at this site here in Utah, put all of them into the field plots first, put the water, drive them down into the ground, set up the water, put in the computer program. The computer program can vary based on the texture and the moisture conditions and what you think are going to be the conditions of the site. There were plenty of times where we had to go back in there and make an adjustment. The machine will tell you if things aren't working right, normally relatively close to the beginning. But for the most part, once you get these set up, they do run on their own. So I'd get these set up and then move on to other portions of the sampling. And then when my timer was up, I'd go in there and move them to the next part of the plot. Big part of our sampling was we needed to take the actual physical soil samples. And so when we wanted to do our primary soil sampling, we did it a little bit different than just sticking a probe in the ground. We dug a hole that was about 15 centimeters by 15 centimeters by 15 centimeters. And we did that by taking a sharpshooter and putting it down straight into the ground on 4 sides, and then kind of going in at an angle and popping out that core. Depending on the root structure and the structure of the soil, sometimes those came out really clean, and sometimes it was a little messy. But normally we ended up with a hole something like this. And then we cleaned off the edges and took a hori-hori or a soil knife and took knife slices. Part of the reason we wanted to do this is because we have 4 different measures of aggregate stability. We know that structure and aggregate stability has a huge impact on soil health, so we wanted to make sure that we were able to get that without having really wet soils that were completely smushed through coring devices. So we dug a hole and we used a hori-hori to take these. And in between each treatment, we did a considerable amount of clean out, because these primary soil samples were split into 6 different bags that got sent off to 6 different labs. Some of that going off for genetic material that Liz will talk about. And so we wanted to make sure that we didn't have contamination between treatments. We also sampled bulk density in each one of our experimental units, and we did this 2 different ways. We had intact cores, and we had ones that kind of went into a bulk bag. We used the same size core for all of them, so we can have an average of 4 different things, 4 different measurements on each plot, in row and out of row. And we basically either used a slide hammer or a piece of wood to push these cores down in the top 3 inches of soil with rings that had a 3-inch diameter. And so we have these intact cores that we'll use for bulk density and available water holding capacity, as well as ones that were put into a bulk bag. All of the protocols that we did out in the field have been put on the Soil Health Institute's YouTube channel, which you can subscribe to. And so I went through kind of what I did in each of these sites and took a video, so that if you have an interest in trying to replicate any of these protocols, they are available. They're also listed, so we'd be happy to answer any questions for you about the protocols that we have.
So now I'm going to give a brief overview of some of the sites that we sampled, as well as the major soil health promoting practices that were at these sites. So I know Cristine kind of showed this already, but I kind of want to reiterate the scope of this project. So we were all across North America. We had sites in Canada, the US, and Mexico. And in the United States, we did divide up the regions. So each of us scientists were in charge of a different region. I was in charge of the Southeast United States, so I had about 20 sites from Texas to North Carolina. Of those 120 sites that we selected, from those we got about 2,000 experimental units, so that's what we called plots. And those plots could vary from anything from really small plots to whole fields or whole farms. Now, these sites varied depending on when they were established. So like Cristine said, almost all the sites were at least 10 years old, but we had about 7 sites that were very historic sites. They were over 100 years old. So it was interesting seeing the scope and how these plots came about. I really enjoyed talking with the researchers. A lot of them were very proud of these sites, which they should be, and it was really interesting to talk with these researchers that actually established the plots. So it was really great working with them. Now, when we first started looking at our plots and looking at our geographical regions, we had this grand idea of making a sampling plan, So we figured that we would start in the south, so I would be the first one to start, and then we'd work our way up north. And by the time that, Charlotte started in Canada, I should be finished with my southern sites. Well, this weather this spring, it didn't really work out that way. So we had to kind of rethink that. So we were really grateful for all those researchers and staffers that were willing to work with our schedules and help us out along the way. So thank you to those researchers and staff for their patience and their time. So looking at the different cropping systems that we have, we can see that corn was the most popular crop that was studied in these systems. And that's followed by wheat. So 72 of those sites had some type of wheat. So that includes spring and winter wheat. Next, we have soybeans, and then a few of the others, cotton, sorghum, and peanut, were some of the lesser studied crops. Now, if you look at these numbers, they don't add up to 120, and that's because a lot of these sites included rotations or multiple crops. So just to give you an idea of where these sites were distributed, we see that for the wheat studies, they're pretty evenly distributed across our continent that we studied. But if we look at something like cotton, it's a tropical crop, we see that most of those sites are scattered along the southern United States. We had a lot of other types of cropping systems, not only just row crops or agronomic crops, but we had some sites that had some type of livestock grazing aspect. So we had a handful of those, and Charlotte's going to talk more in depth about those. So we can see on the bottom left, that's a grazing and watershed study that I sampled in Arkansas. And the bottom right corner, that's actually an integrated crop and livestock system, and that was out in North Carolina. We also had a handful of sites that had some type of tree or forest system. I was fortunate to sample one of those sites in North Carolina. In the top right corner, you can see that's a tree plantation. They also compared tree plantations to successive forests and how that management affected the soil over time. I had one site in Louisiana that looked at sugarcane residue management, and we also had at least one site that had vegetable crops. So this is the distribution of some of our grazing sites. As you can see, a lot of those grazing sites are in the Great Plains of the United States. Now, if we look at the different practices that were examined in these studies, we can see that tillage was very popular, followed by cover cropping. So this is what we see that farmers are interested in and researchers are interested as well. We also had some sites that looked at organic amendments. So that's anything from animal manure to compost to other types of litter. And then some sites also looked at irrigation or water management. So if we look at the distribution of tillage studies, they're pretty widespread across North America. And we see this very similar thing for cover crops as well. And then if we look at which studies looked at both, we saw that about 26 of our sites examined both tillage and cover crops. All right, Charlotte.
So one thing I've learned when I went across my sites, and I said I was working for the Soil Health Institute, I would then have to say I'm Canadian. I am based in Victoria, and I've looked out for the Canadian sites from up there. So just a point of clarification. So there's no data in my presentation. It's picture heavy because they're amazing sites up there, and the principal investigators have these great sites, and I just want to share a flavor of what I saw with you. And so getting at the rangeland sites, the interest was in terms of grazing intensity. So on the day that I went out to Stavely there with the research ranch, somebody was able to fly a drone out. So that was that day, the drone image on the left, and that is with the exclosure. So that's a target, no grazing there since 1949. And then you see the nice outline of it there where 2.4 animal units per month per hectare. So that's the grazing intensity there. And you can see our little quad down in the bottom right corner of that image. And because we were sampling from within the exclosure, that idea of no grazing since 1950, I was able to say, hey, this is where our little plug that Shannon was talking about, how we pulled out that 15 by 15 by 15 centimeter core came out and what it looked like from the zero grazing point. And then at the Stavely area, we were interested in light grazing, so 1.2, and then heavy grazing at 4.8 animal units per month. And so anecdotally, you can kind of see from the two plugs that it looks like there's greater organic matter within the exclosure areas. And then it's harder to see within the heavy grazing, but you could tell there was a lot more bare ground. So that was certainly the target for the rangelands, was the idea of grazing intensity in that sense. And there was also this one from Kinsella Research Ranch, where in the 1970s exclosures were put in and there was moderate grazing around them. And then about 20 years ago that shifted. And so one area, the one on the left, has this exclosure, moderate grazing. And then just upslope from that was the exclosures along with, they went to light grazing. So how does that shifting and how is that change in management practice influence the soil conditions and the soil properties? So that will be really interesting. That drone image was from a previous flyover by the PI on that site. And then there's the landscape level picture there in the corner. I had some summer students assisting me on this, and they were really keen on plant identification. So I sent them off and said, yeah, go ahead, have a look, see what the plants are there. And they said, as they would expect, that's their area. There was greater diversity in the light grazing in terms of the plant community compared to the others. So it'll be really interesting to see what happens there. And then the other point with the rangelands in terms of grazing intensity was also the vegetation type. So this is from 1.4. It's been going since the 1920s. And there's the idea of the exclosures and the grassland, and then you have the different types of vegetation that have been planted. You have wild rye and then crested wheatgrass. So what's the influence of vegetation on the measurements? So we've seen this image in varying forms from the others, so I'll just skip right over it pretty quick just to say that we really have this great breadth of mean annual temperature, mean annual precipitation, so we're capturing a lot of that variability with these sites. So the three that I showed you are in the top there in Alberta. So we're in the 0 mean annual temperature range, and then some of the other rangelands are down in Arizona, so we're into the 18-degree range, and then we go west to east and you hit this precipitation gradient. So if you look at the rangeland sites themselves, they're just this tiny capsule of the overall breadth of the 122 or 1 or 20, depending on what day it is, sites that we have. So going from that, this breadth in terms of the continent, then you get into the soils. And as you'd expect with being agricultural soils, they're dominated by mollisols or chernozems, depending on how you want to name them, or alpha sols or ultisols. And I just like to show the ones that I saw. The one on the far left is from 1.4, the 1920 exclosure rangeland site. And then we have the other 4 representing some of the field crop sites that I had. And that's certainly what these look like. And I mean, it's just amazing, the variety and the wonderful research sites that we had access to and the amazing support that we had out there. They were just so patient with me and getting out there and gaining access across those 3 months of sampling. So we have mollisols or chernozems represented, as I showed, in the bottom left in Alberta. And then, so those are wheat crops, they're wheat rotation. And then north of that, we have the Breton plots, alpha sol here. So slightly different soil type, same crop rotation. So we have a climate gradient there. And then ones, they're Ag Canada Agri-Food, and they are in the south around Lethbridge, and then they go through Saskatchewan, Manitoba. And that's one, there's been a lot of research in those areas of the wheat crops. So we have that amazing research. And then it's going south into the Great Plains. Southern Ontario, we're getting into the corn rotations and horticulture there, which is making links again through the south, and then east to west. So there's great amount of variability. Those are just some of the wonderful pictures from there. So those are the Canadian sites, and I think we'll hear from Mexico next.
All right, very good. Oh, back up just a little bit. All right, so I was asked to give just kind of some preliminary data results on the hydraulic connectivity measurements that we've been able to collect over the past few months. And just wanted to pause for a second and talk about how awesome this project's been, that I've been able to be a part of it, and how blessed I feel traveling around to all these sites, seeing all these cool soils. And as a soil scientist, this is kind of the dream, right? Just to travel around and to actually be able to sample such diverse ecosystems and to meet such cool people. So it's been a real blessing. So what is hydraulic connectivity? Just for a review for those of you who may not work with it on a daily basis, I really like to think of it as the miles per hour of the soil water, right? As the water moves through the soil profile. It's different than infiltration. Infiltration is as the water enters the soil. Hydraulic connectivity is really how it moves through the soil. And there's saturated flow and unsaturated flow. We really focused on the saturated flow of the soil water. So why is it important? Why do we want to measure it? Well, it really water drives a lot of these processes that we're interested in for soil health. And some of these I just wrote down, anything from contaminant transport to groundwater recharge all the way to erosion and ecosystem sustainability. Hydraulic connectivity has been related to all of these different things. How did we measure it? Shannon did a good job of introducing the Saturo. That's a machine that's built by the Meter Group out of Pullman, Washington. Here's a picture of it here that you've seen probably several times already this conference. So it has its own water source, and you see that green box. That's the data logger and the pump, and then it has that stainless steel looking. It's actually aluminum, but a chamber where there's different hydraulic pressure heads with different water and air pressures. And I can, if you want to meet up with one of the scientists on here afterwards to describe or to learn more about the materials and methods of how it works, we'd be happy to do that. It uses something called the two-ponding head method, which has been around for a while. It uses a different version of it, or just a different tweak of it, but it corrects for three-dimensional flow. And then for this project, we did one measurement per plot or experimental unit. And on, I guess, generally or approximately about 850 of these plots have been sampled to date, with plans to sample the rest in the fall. So if each one of those measurements takes somewhere between 60 and 90 minutes, depending on the site you're at, that's somewhere between 75 to 113 days worth of Saturo measurements. And that doesn't count the reruns you have to do if it doesn't perform correctly, or the time setting it up, or transferring it to the different plots. And so it was a big effort going into this measurement. And so we wanted to make sure we were going to get something out of it, and I think we have. So here are some of the preliminary results. So this is from a site in Indiana, Purdue, where they were focusing a lot on the tillage practices. And so you'll see the hydraulic connectivity measurement on your y-axis and the different tillage practice on your x. And you'll see that there's a pretty big difference, a statistically significant difference between the plow tillage practice and the no-till, with the no-till having a lower KSAT value. And if you think of no-till as a sponge, right, it may take in water fairly quickly, but it wants to hold on to it. And so it's not going to flow through the soil profile necessarily as quickly as the till. And there's some conflicting results with that, and so we're interested to dive in a little bit more with this relationship. Again, this hydraulic connectivity versus different hay and pasture cropping system or management system in West Virginia, and the differences in hydraulic connectivity there. And then finally, this is at the University of Missouri Sanborn Field, which is one of the oldest research locations we went to sample. It was established in 1888, I believe. And you can see there's quite a difference between the hydraulic connectivity between a monoculture and then a management system with a crop rotation. So, and really, in conclusion, we know it's a preliminary sketch, but with all that time and effort going into the hydraulic connectivity measurement, we're really excited that we're starting to tease some of these relationships out, and it gives us just some more excitement about the further analyses that we're going to have between this measurement and other indicators and at other sites. And then lastly, just here's some pictures. You'll notice that we went through a lot of FedEx boxes. That's just one site worth of FedEx boxes in soil. And then all these different site PIs or principal investigators, technicians, and I even got to bring my family along with me to sample some of these sites. Paul joked maybe there's some child labor laws involved here, so I appreciate your discretion. Don't turn me in, but we did take our daughter with us, and she helped. It was great. So with that, thank you.
I wanted to visit with you really quickly and give you an update on the progress that we had at our Mexico site, and let you know where we are in that process. The first thing we wanted to do is, since we didn't have a lot of contacts in Mexico, is during the application process, do a very thorough job of evaluating the applicants and see if we could find a partner that could help us down there. Logistics, time, funding for us to be down there for 3 or 4 months in a row was just not going to work out with this project. And we were very fortunate in that we found a team from Summit that was willing not only to help us with their sites, but also help us with all the sites in Mexico from 5 different organizations. They provided us a lot of transportation, but also sampling during the times that we couldn't be there. There's a network in Mexico. It's called Masagra Network, and it consists of several hubs consisting of about 58 sites throughout the country. We selected all of our sites from this network. It just played into what we were trying to do with this project perfectly. And Summit organized the other organizations that were part of the network, and they were our primary site providers, but there were also several other organizations that helped us with this project. The sites in Mexico were very well dispersed, ranging from northern Oaxaca up to southern Sonora, encompassing most of the agricultural areas in the country. And so we think we have a really, really good footprint down there in terms of the sites that we selected. We went through the 58 sites from the Masagra network and selected the 16 that are part of our project based upon similarities, not only with cropping systems, but with the time. Several of their sites had not been in production for very long. So this is kind of the breakout of the 16 sites. All 16 of them had a tillage component to them. And some of those were raised beds, and some of the raised beds were permanent semi-no-till, and others were tilled every year. Residue management in Mexico is very important, and it's a management practice that they're trying to change. There's a lot of burning that goes on, and there's a lot of residue that's removed from the fields, either for animal feed or for bedding. And removing that residue, we think, has a very negative effect on the overall soil health. And so we wanted residue management or retention to be a key component of the sites that we selected. And then crop rotation was a major treatment within 11 of those sites. We wanted to make sure that we were uniform across all of our locations from Canada to Mexico. And so Daniel Liptzin and myself went down there for a couple of weeks, and we trained the Summit crew on the sampling protocols that we wanted, that we were using at the rest of the sites. And we also sampled 4 sites with them. And then we left some equipment behind, and they sampled the other 12. Now, that phase of the project went fairly smoothly, with the exception of one thing: they're vertisols, and only a Texas girl could love vertisols. I heard that earlier today. They're really tough to work with, especially if you're running water measurements and you have cracks that big and they're dried out during the dry season. So that was about the only limitation to the sampling that we had down there. Everything else went really smooth. Had some other issues with processing. Wasn't too bad when we were close to Texcoco, which was Summit's headquarters. Maybe a couple of delays in getting the samples back to headquarters when we were out 8-10 hour drive. But shipping did present us some problems. We were going to 5 or 6 laboratories in the U.S. from 16 sites in Mexico, and the customs and the quarantine folks at the USDA changed some things around in January, and so we did have some packages have some issues. Dan on our team was, I call him Mr. FedEx. He was somehow got all the soils back in a reasonable time, so we were really, really happy with the way that ended up. But there were some frustrations getting all these soils back across the border. Had a 5-minute talk, and you can't really talk about data too much in 5 minutes, but I could not resist myself. We only have the Satura data that we have information for, and Max showed some of that. Just wanted to show you one of the sites from Mexico. If you look at the two bars on the left, those represent a wheat-corn or a corn-wheat rotation. We purposely only chose 1 year within a 2-year rotation for most of our sites, but this site we sampled during both years, and it's really nice to see under conventional till with a raised seed bed and residue retained, there was no difference in sampling of those two. And so that tells us that we made the right decision there. If you move those same two treatments up to no-till, we did get, opposite of what Max saw in Indiana, we did get an increase in saturated hydraulic conductivity. And then lastly, when we removed the residue, we crashed our water infiltration or our water movement numbers. And that's something that is very important for what they're trying to do in Mexico with their residue retention. I'm really excited because we're just starting to get this data to trickle in. And our Mexican colleagues are also very excited to see this because the data that we're going to provide them should do a good job in helping the Masagra network to get more farmers into their programs. Thank you.
So I'm Dan Liptzin, and my job was to talk about soil organic carbon and enzyme dynamics. And as I was thinking about what I was going to say today, I realized that I should have given it a different title. So I'm really going to talk about chemical signatures of microbial processing of carbon and nutrients. And so the indicators I'm going to be talking about is a subset of what Cristine mentioned. We had about 30 of them. And these are the ones that are the chemical residues left behind by microbial processing of plant residues in the soils. So the specific indicators I'm going to talk about are soil organic carbon, the active carbon that we measured with permanganate as permanganate oxidizable carbon, 4-day carbon mineralization rates, the soil protein index, nitrogen mineralization, extractable phosphorus, and a suite of enzyme assays. And the different colors indicate what type of indicators these are. So the blue ones are the amount of this indicator, so at the total pool of this indicator, and then the white ones are measuring some rate of some process related to microbial activity in the soil. So I'm a biogeochemist by training, so I don't think in pictures, I think of boxes and arrows, so I apologize for that. But so I just was going to run through quickly the carbon cycle and nutrient cycles so you can see how these fit in to those systems. So in these agricultural systems, like all terrestrial systems, there's plants in the system that take carbon dioxide out of the atmosphere through photosynthesis, create leaves and stems and roots. And so where we're really interested in is how the management practices in these systems shown by the tractor affect how that carbon gets into the soil. So some of that carbon is removed to be harvested, some is left behind on the soil. And so one of the things we're interested in is how this management affects that black box, the total amount of carbon in the soil. Another indicator we're looking at is the active carbon in the soil. So this is just a small fraction of the total carbon that's readily available for those microbes to decompose. And then thirdly, one of the other activities of microbes, besides just breaking down carbon into other forms in the soil, is also to produce CO2. So one of the results of their metabolism is to turn carbon in the soil back into carbon dioxide in the atmosphere. And so we can get a sense of the rate of that by measuring the carbon mineralization rates. So we're also looking at a bunch of indicators of nutrient cycling. So once again, the source of a lot of these nutrients is the plant residues coming into the soil. There's a big function of the management once again. So are they fertilizing with manure or with other types of fertilizer? And so there's this pool of soil nutrients in the soil. And we're not measuring all of the pools of these soils, but we're measuring a few of them. We're measuring the soil protein index, which is the amount of what should be a readily decomposable source of nitrogen that should be readily available to plants. We're measuring the exchangeable pool of phosphorus, which once again should be readily accessible to the plants in the system. And then we're also measuring some rates. So we're measuring the nitrogen mineralization rate, so how quickly those microbes are converting the organic forms of nitrogen into inorganic forms that are available to the plants. So once again, measuring some pools, some fluxes of these nutrients in the soil. And then finally, we're looking at a suite of extracellular enzymes in the soil that also give us some sense of the rates of these processes in the soil. So we're looking at 4 different enzymes, one that's more associated with the carbon cycle, one that's with the nitrogen cycle, one with the phosphorus cycle, one with the sulfur cycle. And these extracellular enzymes are excreted by microbes into the soil because they increase the rates of reaction in the soil. So all of these enzymes are specific to certain substrates in the soil. They're just a subset of all of the possible enzymes in there, but they're relatively stable and they're possible to do on relatively dry soils, are less sensitive to the moisture content of the soil and can be even done on dried soils. And so the exciting thing about all of these is they're really good indicators on their own, but there's also the possibility that we can look at the ratios of them. We can look and see what the coupling is like between carbon and nutrient cycles in the soil.
Hello, I'm Liz Rieke, and I'm going to give a brief overview of, there we go, of the genomic indicators. So we're 3 genomic indicators that were included in the project. First of all, I'd like to thank Dr. Knabel for his great talk this morning and the work that it was great to hear about the work that ARS is currently performing on the microbiome. We're really starting to scratch the surface with identifying the structure and function of these different microbial communities in soils. So the first indicator that I'm going to talk about is targeted 16S rRNA targeted amplicon sequencing. So going back to Shannon's talk last night, really thinking about that, how many bacterium are in 1 gram of soil? You're ranging from over 1 billion to tens of billions in just 1 gram of soil. So each of those bacteria contain the 16S rRNA gene. And within this conserved gene that each of these bacterium have, there's variable regions and conserved regions. And so based on the variable regions within them, we can get down to the genus level identification of these bacterium. So with that, we target this variable region within this gene, and we amplify that variable region. And from that, we take it and match it to a library or database of its taxonomic identification. And so from there, we can kind of map out the different relative abundances of these within your different biomes. So that's for the bacterium. Besides that, we do somewhat similar idea. We're actually amplifying a region for the fungi that's in between 2 of these different ribosomal genes. So same kind of idea. It's this variable region that we can get down to that genus level to really kind of get a deeper idea of what's going on in these. So if you go back to some of the other indicators that have previously been used, such as the phospholipid fatty acids, you really you can look at your ratios of your fungi to bacterium at kind of a higher level. But with these, we're really going to be able to dig down and look for hopefully a small subset of these different genera that we can tie in to management practices, and also these other different, the other 30 different indicators in hopes of finding some microbial indicators of soil health. And so the last genomic target that we're looking at, which some of you may be familiar with, is shotgun metagenomics. So instead of trying to target this one piece of this gene that's conserved between these different types of soil microbes, basically what we're doing with the shotgun metagenomics is taking your DNA, your total microbial community environmental DNA, and putting it through basically the equivalent of a paper shredder. So we shred up all the DNA into these small pieces and then work to reassemble this puzzle. And within this puzzle, we have other libraries where we can match either taxonomy or different groupings of functional genes. So some of the different groupings of functional genes that we can look at are nutrient cycling genes, genes that confer pathogenicity to either humans or your cropping system. And then also antibiotic resistance, which Dr. Noble touched on briefly earlier. So between the shotgun metagenomics, looking at the functional identity of some of these different cropping systems, and then the taxonomic IDs, we'll really be able to get a deeper understanding, I believe, of what's going on under there. And with that, I'll hand it off to Michael Cope.
Hello, my name is Michael Cope, and I'm going to give a quick update on the content and structure of our project database. Here is a graphical abstract of the data model for our project database. A data model identifies elements of data in the database and defines and standardizes how they are related to one another. We've heard a lot today about soil health measurements. I think most of the people in this crowd will understand what that data physically looks like and what it contains. We are or will be downloading 10 years of daily weather data from weather stations at experimental sites where that information is available, but also through DAYMET, D-A-Y-M-E-T. DAYMET is an interpolated weather data set that is sort of modeled and coerced to be spatially and temporally complete at the daily scale for North America. We just heard some about soil health genomics. This data is so conceptually and structurally different from the other measurements. It requires its own storage and management and analysis strategies, so we are treating it as sort of its own separate entity away from the other measurements. But then there's the project metadata. I'm going to go into a little bit more detail here in the next slide, but our data model must state how these elements are related to one another, and for our project, these are related to one another through an index that we created for experimental sites, experimental units, and samples. This is really how we manage data from the field to the labs and back to our database on our servers. And then also the spatial temporal index, which is critical for database queries and building in scalability into the database, but also retrieving information from other data sources like soil geographic databases, weather databases, or integrating databases with partnering organizations. With that, we'll take a closer look at another data model that is dedicated specifically to the core of the project metadata. This is still general and in progress, but it is more like a traditional SQL data model. In blue, here are each of these boxes are tables or spreadsheets or data frames. In blue, at the top, are the table names. Italicized above these dashed lines in each table are column header names that are for design join keys, and then not italicized below the dashed lines are example or actual column names for that contain information that are unique to these tables. And these black lines generally indicate how one might go about constructing something like a select and join operation to query and retrieve information across these tables. There are 3 join keys that are hierarchically common to these tables: the site ID, the EU ID, the experimental unit ID, and the sample ID. And down here in the lower left is an example of what those look like. So the general gist of it here is that we have a list of experimental sites, and we have some information that is unique to those experimental sites. Then we have some experimental units at those experimental sites and some information that is unique to those experimental units. Then we have some samples from those experimental units and some information that is unique to those samples, and so forth. And then we can break down here and go into management practices, where we have a management practices timeline for each treatment or experimental unit ID, and perhaps some very specific information about actions in management practices. So why is it important to think these things through and declare our data model? Well, the most obvious reason is so that we are organized. We know what information and data is where, so that we can quickly retrieve it, inspect it, and then eventually analyze it. I'm working with all of the scientists here to finalize their metadata for all of the sites that they sampled, but I've been working specifically with these sites in Mexico. And here I have this information loaded into a geo database in ArcGIS Pro, and I'm telling ArcGIS Pro just quite simply count experimental units at these sites by these generalized treatment names. And so that is my update. Our work continues. Thank you.
All right. Thank you all.